Hello there and welcome to Bio 138. Today what we're going to be taking a look at is just some basic chemistry. We're not going to be going into some of the chemistry maybe that you did previously, like in a high school chemistry class or even a high school biology class, but instead we're going to kind of get you the functional stuff that you happen to need that's going to serve as the foundation here of the course as we move forward. So with that, there does happen to be quite a few objectives that we're going to be taking a look at here today. And more than likely, what I'm going to do is actually divide this up into two different PowerPoints, just so that way you have the ability to stop this and you can go back to what you would like to view and everything. And so more than likely, what I'll probably go ahead and do is, I'll probably say that this right here is going to be kind of chemistry number 1 objectives for the PowerPoint, where we're going to take a look at organic molecules and everything else. And then from that point, this is going to be chemistry video number 2. Looking at, again, the laws of energy, looking at some of the different types of reactions that end up occurring and how enzymes come into play here, as well as the pH scale. So that's going to be what probably video number 2 is going to be. So I'll break them up into 2 videos there for you. Otherwise, like I said, this can be rather a long video if I do it as well. So just kind of letting you know about that. So with this first video, what we're going to take a look at here is what we call organic chemistry. And we'll kind of talk here about this important molecule, which happens to be carbon. Carbon serves as the functional backbone, if you will, that gives us all of the other building blocks, if you will, for life. So it's a really important atom, and I'll explain that to you actually in just a few moments. And it allows us to build a lot of different molecules. And so it's, again, a very important atom when you take a look at it. Okay? And then, like I said here, we'll go through some of those different building blocks here, like carbohydrates and fats. And then we'll also go ahead here and take a look at proteins and nucleic acids. So that's what we're going to look at for this video. Okay? Now, as we go ahead here and move forward and everything, many of you have taken some sort of basic high school chemistry class. And so, when you take a look at, like the structure, if you will, of the atom, right? So you happen to have your atom, and you've got the first part of the atom right here, which happens to be the nucleus. And if you happen to recall the nucleus, you've got some protons in here. And then you also happen to have some neutrons in here, okay? And then located outside of the nucleus is going to be what is known as either shells or orbitals. And this is where the negatively charged electrons are going to be at, okay? So that, again, happens to be the electron, all right? And this is going to be the shell. And if you know anything, again, from basic chemistry, when you start taking a look, so if I kind of take this diagram here now, and if I only show you this first shell here, there can be a maximum of 2 electrons here that can go into this first shell. And then it's completely full. Once this shell is completely full, there's no other electrons in here, and thus there's no other way for this atom to go ahead and bind, or if you will, bond with anything else. Well, again, taking a look at your number of electrons right here, this would show you what helium would be. But now, there of course happen to be atoms out there that have more than just 1 or 2 electrons, and so you happen to have additional shells or orbitals that go away. And so the next shell over here can contain up to a total. So we've got 2, and then right here we've got 1, 2, 3, 4, 5, 6, 7, and 8. So this next orbital right here, when you start taking a look at it, can have up to 8 electrons. And so this gets to be really big when we start looking at bonding, because what's unique about carbon is that carbon is going to have 6 electrons. And so with those 6 electrons, again, when you look at the very first shell that we happen to have right here, the very first shell can take 2 electrons. And again, I'm just showing you the electrons here by putting in these dots. Well, obviously, if that first shell can only take 2 electrons, if I just take 2, right, minus 6, I get a number here of 4. Well, how many electrons can I put now into the second orbital? I can place now 4 electrons here. And so when you take a look at carbon, carbon's unique because now you can actually put an extra electron somewhere in here. You can put another electron over here if you wanted to. And then you can also go ahead and you can put another electron right here. And then you can also place another electron right about in here. So the nice thing about carbon is that carbon can form 4 additional bonds to fill up this outer shell. Now again, if this was a high school class, we would want to talk about how atoms would like to, again, stabilize their outer shell by filling it with electrons. And the only way that you can do that typically is by bonding with other atoms that happen to have electrons that are available. So if this is our atom right here, that is carbon, the nice thing about carbon, again, is that 4 atoms here, or I apologize, these 4 electrons here that are available, you can bond with, like, let's say, another carbon atom. So you could go ahead here and you could fill up this little orbital area by having another carbon atom come in here. And now I'll draw a bigger dot right there, and I'll make that dot bigger. So again, now we're going ahead here and we're sharing these electrons. And because we happen to have this sharing of an electron going on, we have a type of bond that is going to be known as a covalent bond. And there still happens to be another electron right here, another one over here, and another one over here. So if this again happens to be a carbon atom, we could do another bond with even another carbon atom. And so the unique thing here that we start running into is that we can start forming some really big chains of carbon to carbon. And for those of you that have ever taken a chemistry course before, this right here happens to be a covalent bond where we're actually going to be sharing an electron. And that's exactly what I have drawn in here, showing those dots together there. And so now, because we have these covalent bonds that are occurring between these carbon atoms, we can get a whole bunch of different shapes. Because what you can kind of think about now is that this is kind of like having a Lego set where you're now going ahead and you're attaching, different Lego pieces together. So, we could go ahead and we can have a covalent bond here, and then we can even go ahead and we could put like a hydrogen atom over here. And, if I come over to this diagram, hydrogen happens to have, again, only 1 electron. So we could go ahead and, again, we could do a sharing right here between this area. And so, again, that would be another covalent bond that can form. And so we can start building these big, what we call organic molecules. And organic molecules, they're going to contain carbon. This is the key to it, okay? The only time that a molecule is not organic is when you happen to have things like carbon dioxide. This is going to be an exception to that rule, where if you look at the chemical formula, you'll be like, well, there's carbon there, so it must be organic. Well, this is the exception. Carbon dioxide does not happen to be an organic molecule. But if you look at this formula, C6H12O6, this happens to be the chemical formula for a very important sugar that is known as glucose. And so we'll be talking about sugars here in a little bit. And if you notice right there, there is carbon, and so thus this glucose here happens to be a organic molecule. Okay? So just as an FYI there for you. Okay? Now, the big thing, like I said here, is that you can go ahead And if you notice, like there's a number 1 and number 2 and number 3 here, think of these as being atoms, if you will. And so what you can now begin to do is you can put these atoms together and you can build like a little short molecule that we call a polymer. And then as you begin to put more and more atoms together, so if you notice right here, this happens to be another little purple one. Well, if we start linking that together, we can go from building what is known as a short polymer, and we can get this to be a much longer polymer. Well, poly again happens to mean many, and so the way that you end up building a big polymer is by putting together little subunits right here that are known as monomers. And so these right here happen to be the very small building blocks. And so that's what a monomer happens to be. And so we're going to go ahead and take the monomers and put them together to build a polymer. And as we talk about organic chemistry, there's going to be very specific monomers, very specific building blocks for each of the organic molecules. Okay, so they're going to have some different names here. But all we're trying to do here is link together one or two of these pieces, and then we start putting them into a longer channel, if you will, or longer chain right there, known again as a polymer. Okay, so that's what we're going to look at. All right. Now, when we start talking about these organic molecules, they're what we call the macro. And so if you ever take a look here at this prefix, macro, of course, means this is going to be a large molecule. Okay? And there happen to be 4 different types of macromolecules. And if you think of your diet, as far as in general terms, like what are the food groups you eat, and we're not just talking about going ahead and eating pizza and beer and things of that nature. But if you think about your main 4 food groups here, you're going to have, your carbohydrates. And then, of course, your proteins. And then the other one that you're going to have is going to be your fats. And then the one that really is not a food group that's necessarily for any sort of energy storage, for long-term energy storage, is going to be one that we use to pass on genetic information, and that is going to be known as the nucleic acids. So as we look at the macromolecule groups, there's going to be 4 of them, and these are the 4. Okay? And again, going back to this slide over here, those 4 main macromolecule groups here are the polymers. And like I said, each polymer is going to be built from a monomer, a single small building block. And again, each of those groups will have their own building blocks. Okay? So, the first one that we're going to take a look at here is the carbohydrate. And the carbohydrate, when you really start taking a look at them, their monomer is this right here. It's going to be called a monosaccharide. With that monosaccharide, you've got the basic chemical formula here of C and then 2 hydrogen atoms with an oxygen. This right here is going to again be the carbon, and like I said, carbon gives you that backbone. Okay? What do carbohydrates end up doing here for you? Well, they're going to go ahead and they help you with storing of energy. And so a lot of times that's going to be in the form of some sort of sugar. And one of the ways that you know you happen to have a sugar is a OSC ending. So if you look back over here to this slide, notice right here how I have the word glucose, and notice again how there is a OSC ending. This again happens to be a sugar. So again, sugars happen to have OSE types of endings. Now what we're going to go ahead and do here, the way that we build these carbohydrates is we take this monomer, of a CH2 and an O. And again, we start linking them together to build a polysaccharide or to build a big sugar. And so looking at this drawing that I happen to have right here, this happens to be glucose. This was that sugar that I just told you about. And so like I said, the chemical formula is C6. So you can see the 6, again, sugars that are in here. I apologize, the 6 carbon atoms that are here. And then you can see the hydrogens that are breaking off from here and the hydroxyl groups, if you will. And so this again happens to be that molecule of glucose. Now some of you may have actually seen this sugar before. This sugar happens to be called fructose. Fructose has the exact same formula as glucose. So if you notice, there's 1, 2, 3, 4, 5, and 6 carbons in there, okay? But the difference is the arrangement of all of these other molecules. So if you notice on this 3rd carbon atom coming down, how we've got this hydrogen hydroxyl group over here, and notice over here how they happen to be switched around. And then the other part right here is on the 2nd carbon, we've got an oxygen, whereas over here we happen to have a hydrogen and what is known as a hydroxyl group. So you can see there's some differences here. And this is why, even though they've got the same formula, they are different sugars. And so if you happen to know anybody who is a diabetic, sometimes you'll have individuals who are diabetic and they still want like a cookie, but they can't really take this glucose over here. They have to take a different sugar. So sometimes they use cookies over here that are made with fructose types of sugars. And go ahead, go to a large grocery store, and there's going to typically be like a health food aisle, and they may have some cookies over there known as Frookies, which actually contain fructose. So give them a try sometimes. They do taste different. I definitely will tell you that they've got a different taste to them. So give them a shot sometimes. See if you can't taste those cookies, because they do happen to be unique when you take a look at them. Okay. Now, the other things that we start getting into, I mentioned again, energy storage, right? That's what these molecules end up doing. Well, what you can start doing is you can take even. So if you can notice over here, this is my glucose molecule. And what you can start doing, oops, wrong way, I apologize. What you can start doing is you can actually take a glucose. So when you look at this little diagram right here, this diagram that I happen to have right here does happen to be a glucose molecule. And so with that, you've got a glucose that's going to be chained to another glucose, which is chained to another glucose. And so what you can start doing here is you can start building molecules like starch. And if you've ever had starch before, like in a potato or something of that nature, right? Potatoes are a source of energy. And so that's why sometimes people say to watch your starch intake, because again, you're taking in a lot of sugar molecules, because again, you've got this glucose molecule that's linking together to make this, starchy type of molecule. In humans, we end up making, and other mammals too, we make a molecule to store energy known as glycogen, and then we end up breaking this down inside of our mitochondria, and that allows us to make a molecule known as adenosine triphosphate. And so we can store energy in the form of that glycogen right there. So, and again, when you start looking at these, unique little structures right here, and again, I am the world's worst artist, but that right there, that would be the glucose molecule. And like I said, they're just chaining all together to make that glycogen. And so that's going to, again, be the molecule that you know as glycogen. Okay. Now, if you want to, you can still take this glucose molecule and you can start really putting it together. And then the other thing you can start doing is you can begin to twist it and everything. And so you can get that glucose molecules to now kind of form a chain and begin to twist. And again, notice right here, that as we go through this molecule and it begins to twist and everything, we form, so notice right here the OSC, we form a sugar molecule called cellulose. Now this is really a really complex molecule, and it's really hard to digest cellulose and break it down, and this is actually what paper fiber is made from, if you will. So it's made again from the plant cell wall, and you don't have a way to digest it. And if it's the summertime and corn on the cob is there, and let's say that you have a couple of ears for dinner, and you wake up and you, for breakfast, have a couple of ears of corn on the cob, and then you happen to have some for lunch, and then you have some for dinner. You wake up about 2 days later, you use the bathroom, you look in the toilet, and you go, oh my gosh, there's corn on the cob. Well, why didn't you digest that? Well, it's because of all this cellulose. It's a really tightly wound molecule, and you don't have the digestive ability to break it down. So that's that cellulose. So back to it, the slide over here, when I talked again about, what is it that carbohydrates do? They store energy, but they also help with structure. So they are also going to give you structure and support. Okay, so those are some of the roles there of carbohydrates for you. Okay. The other molecule that we happen to have here is going to be a lipid molecule. And when we start taking a look at a lipid molecule, right, what is the monomer? The monomer is actually going to have two parts. It's going to have this right here, which is called a glycerol group. And then the other part that they have is they have a fatty acid tail. And the nice thing about lipids and everything is that this is going to be typically where you store energy for long term. The other thing too that you also have with lipids is you also have things that are known as triglycerides. You'll have steroids, and then you also have oils here that will come into play here as well. Okay. With this again, lipid, what again you'll end up seeing is that glycerol group with that fatty acid tail. And when you start taking a look here at again saturated versus unsaturated fats, what's the difference here between this plant fat, if you will, versus this one over here that has animal fat? Well, they both happen to have that glycerol group that you happen to see right there, but the difference is that this plant fat right here happens to have an additional covalent bond right here that gets this to bend. And so because of that, this unsaturated fat typically is going to have less issues than if you're eating a diet very high in animal fats and things of that nature. Because again, this one right here is so big, it's just going to go ahead and get stuck a lot easier than this one over here. I don't know if any of you guys ever tried these before, if you ever noticed this right here where it talks about oleic acid. There was a time when people wanted to have like a quote-unquote healthy potato chip, and so they had what was known as like an oleo chip, and people would go ahead and eat them, and some people would get really gassy with them. So if you ever know anybody that tried some alternative types of potato chips and got really gassy, they were trying some different types of oils and everything just as part of a health food craze, and people were really gassy with them. So you really don't see them on the store shelves. It's been a while since I've seen them. So just as an FYI there. So, okay. The third type of molecule is a protein. Now, proteins are kind of unique molecules. They're pretty complex when you start taking a look at them, and the nice thing about them is they're there for structure and support. And then the other thing that they do is proteins also form a type of molecule known as an enzyme that we'll be getting into with the next lecture. Okay? The monomer, or the building block for a protein, is known as an amino acid. And with the amino acid, there happen to be 20 different types of amino groups altogether. So there's actually 20 different forms of this monomer. And this right here actually would be an amino acid that you see right here. And the way that you end up getting different amino acids is by actually taking this little R group right here, and this is where you change out this little part of the chemical, and then you put in a different R group here, and then you get a different amino acid. And if you do that in 20 different variations, you can get 20 different types of amino acids. So that is, again, the protein molecule. Now, what's unique here is how these amino acids begin to bond together. So if you remember again how I mentioned this previously, how you take like a monomer and then you add another monomer and then you add another monomer, and eventually here you're building, again, a polymer. And again, poly means many. When we're actually going ahead and building a protein molecule, we'll use the appropriate terminology here with what the polymer actually is. But again, the building blocks, these little circles here, are these amino groups that we're putting together. And then when you look at this whole thing here, this is going to eventually go in to the polymer that you know as being the protein. Now, the unique thing is that there's actually 4 steps to this process to build the actual polymer, if you will. So if you notice right here how I have this molecule, this actually is one amino acid. And now we're going to attach a second amino acid. And so you can see here how you got this thing right here, GLY. This is glycine and isoleucine. And now you can see how they're put together. And then we're going to bring in a valine over here, and you can see how they're linking together. Well, we don't just call this a polymer, but we're going to call it the very specific form of polymer to a protein, which is known as a polypeptide. And normally this is what we call the first level of protein structure, that we're going to put together amino acids. Okay? Now, eventually what's going to happen is as we begin to put like hundreds of these together, imagine, if you will, that you happen to have a bunch of preschoolers. You put them out in a hallway, what do they begin to do? If they were in a straight line, they're going to want to go ahead there. They're little kids and everything. You never know, did one of their dads give them a Bang energy drink before they came into school, and now they're all jacked up, like the kids on Talladega Nights, like, again, they're just kind of all crazy and everything. So what happens here is they begin to move around. And so what happens is you begin to bend and your line begins to twist and everything. And so these amino acids begin to bend and twist because of what actually happens here. Like if you notice these couple here that are really close together, these over here begin to form some bonds between themselves, and that's why these shapes begin to occur. So this is what we call the secondary level of protein structure, when these amino acids now begin to cause that straight line to curve and to twist. Okay? After we get from that first level, which is known as the primary, and this again happens to be known as the secondary level right here, and so you can see these bendings and twists and everything else, we start getting into the next level, which actually happens to be known as the tertiary level. And so what happens in that tertiary level, we get additional bonding in here. And when we start getting some additional bonds in here that start forming and everything, what starts happening is that we're going to get a whole lot of bending that's going on. And so then finally what we end up doing after that tertiary level, we get to the final level, which is known as the quaternary level, where we actually take some different groups of tertiaries and put them together to make this big, huge blob that you happen to see here. So this again just happens to show you the 4 levels again of protein structure, where again you've got this over here, which is going to be the primary, the first level, and then this right here happens to be the secondary level, and then over here happens to be the tertiary, and then the quaternary level. And what are proteins when you look at them? A lot of times we're going to call them a glob. And so in many cases, we'll say, hey, this is globular, that proteins are a big old balled up mess, if you will. And if you actually end up taking like an immunology class We use the proteins too to make up different types of what we call antibodies and things of that nature. And those antibodies are big globular proteins. So sometimes we use terms like immunoglobulins and things of that nature. Just we've got this big balled up mass of protein, if you will. Okay? So that is a quaternary structure. Okay? And so proteins are pretty complex. And as I mentioned to you, they give you structure, they give you support, and then again, they also form what are known as enzymes. And enzymes speed up reactions. And like I said, we'll get to that here in just a little bit. So you can kind of see right here how this ends up going. Okay? So you've got these little amino acids, and then you've got this line that begins to twist and bend, and then you get some more twists and bends, and then you take these big globs and you kind of put them together. Okay? Now, the other thing that can happen to proteins is that they can be broken down. They can be denatured. And so what is denatured? This is when they're going to go ahead and the shape changes. And there's several things here that can change that shape. And what really actually happens in here is that the shape changes because you break bonds. And so when you break bonds, things begin to fall apart and everything. And so examples of things that can go ahead and cause this to denature, well, you can have things like temperature. You can also have things like pH. And you can also have things like salt that cause that to go ahead and denature and break down. And so when you take a look at this area here, these are all things, again, that break down a protein. Well, if you think about it, right, protein happens to be like a meat product, if you will. Well, let's say that we're going to go ahead and we're going to season, some meat, if you will. And so sometimes we have these things that we call marinades, right? And if you think about a marinade, right, what is in a marinade? Well, in a lot of cases, if you actually, open up your fridge and you take a look at it, maybe you have Italian dressing, or you go ahead and you buy like the Lawry's variety of, not seasoning salt, but Lawry's makes marinades. And if you actually look at them, a lot of cases they're going to have different types of acids. And the acids, there is a pH, and what that does to your meat, it actually makes it more tender because it breaks down these bonds and everything. And then the other thing that some of these marinades have in them is they also happen to have salt. And if you've ever heard of salt being used as a tenderizer, that's what it ends up doing here. It breaks down these bonds. And then what do you do with your meat? You cook it. And so what does cooking end up doing? It's going to break that down and it's going to denature it. Now, here's the key part to these bonds that you end up breaking. If you only add just a little bit of pH, these bonds will want to go back and repair themselves, if you will. It's really not a repair, but they will come back together. But if you add way too much pH for a long period of time, it's possible for you to completely break this down. And so, again, you do happen to see that from time to time. Okay? So, like right here, of course, happens to be the actress Nicole Kidman. This photograph is now probably over 20 years old. But again, when you start taking a look at bonds and things of that nature, for those of you that happen to have curly hair, you can use a flat iron to straighten out your hair. And so what's happening there is you're actually going ahead and you're breaking these bonds so that way the hair is not as curly. And like I said, if you don't add a whole lot of heat, those curls will come back because those bonds will reform. Now, if you add too much heat where you burn and singe your hair, the hair is not going to go back, but on the same token, it's now singed, and so thus it's damaged, and again, you probably don't want to see that. So that's, again, the difference here, between these photographs here, that you can denature these. The other important thing is this diagram right here. Notice right here how I've got the 1, the 2, the 3, the 4, the 5, the 6, and the 7. Well, this right here happens to be the red blood cell, right? And red blood cells, again, they carry oxygen. And again, they also carry carbon dioxide. And so they're responsible, again, for getting the oxygen to your cells and taking the carbon dioxide away from your cells, and again, allowing you to get rid of that. Well, when you take a look at this specific amino acid right here, right, going back to the importance, why is the sequence of amino acids so important? It's, again, kind of like putting directions together and not putting them in the right sequence. Well, if you put in the wrong thing, you get the wrong types of bonds. And so this over here happens to be known as sickle cell disorder, or sickle cell anemia. And so what happens is that just because all of a sudden the wrong amino acid, and it's only one amino acid, that's it, just one, out of several thousand, is put in the wrong place, rather than getting these nice little red donuts right here, you get this kind of sickling moon. And some people have this disorder and they function fine throughout life. They don't have it very severely. But some people get it where they're incredibly severe, where they have a lot of these sickled cells, and so they suffer from a lack of oxygen. If you happen to watch NFL football, there is a commentator, and he used to be a safety for the Pittsburgh Steelers, and his name is Ryan Clark. And whenever Ryan Clark and the Pittsburgh Steelers would travel to Denver, Ryan Clark could not play in Denver. And the reason why is, Denver's known as the Mile High City. And so when you get an elevation above a mile, you happen to have less oxygen there. So because of his disorder here, because he had sickle cell anemia, he was not able to play because of the risk that if he ran out on the field and he was sprinting, there may be the chance that, at some point where he might not be able to bring enough oxygen into his body, in which case there might have been some brain damage potentially. So this is kind of one of the quote-unquote home field advantages of the Denver stadium, is because, again, it took a player like Ryan Clark, who's actually a Pro Bowl safety, and he couldn't play in that elevation. So, and again, with him, all that was wrong is just one of these amino acids. And like I said, there's 20 of them. And so he just had the wrong one in that place right there. So these are pretty big things when you take a look at them. So again, these happen to be your macromolecules. Okay? Now, the fourth group right here are known as nucleic acids. Okay? Nucleic acids are really here more for your genes, and this is how you pass on your genetic information. Now, remember again how I was using the word monomer, right? What is the building block? Well, the monomer of a nucleotide, a monomer of a nucleic acid is known as a nucleotide. And there's actually going to be, when you start looking at nucleotides are going to have 3 subunits. They're going to have a sugar molecule, they're going to have a phosphate, and then they're going to have something known as a nitrogenous base. You're going to really want to know this for the future, because as we go through this class here, we're going to talk about nucleotides in the future with genetics, because again, this class is a class about, heredity and ethics and everything else. Well, you got to know that molecule, okay? If we change this molecule right here, the sugar, we get some different forms of nucleic acids. There's one that's known as ribonucleic acid, where we use the sugar ribose. And then there's one that's known as deoxyribonucleic acid, where we use the sugar deoxyribose. And as I mentioned here later on, we're going to go through this whole process on how your body eventually is going to take this genetic information and go ahead and actually build a protein through a process known as protein synthesis. So that's going to be in the future here. Not going to get to it today, but that will be in the future. And this molecule, like I said, will come back again. Okay. And so again, taking a look at this structure right here, this right here again happens to be the nucleotide. And so right there is the sugar molecule. Right here is the base. And then right there is that phosphate group. And so you can see that little letter P right there. That's a phosphate. The important thing is this right here, the way that we end up going ahead here and we change what protein you end up getting built is by changing the base. So that's what we're going to change up in here. Okay. Otherwise, again, when you start looking at this molecule, like if you look at 1, 1, and 1, they all look the same. If you look at 2, 2, and 2, they all look the same. If you now take a look at 3, 3, and that 3 right there, notice that one right there looks different. And so it's the bases right here that we change that gives us different types of genetic info. And so again, we're going to be going ahead here and we're going to be building different genes from this. Okay? So that's what we have there. Now, here's what's going to happen. The other molecule that we also need to talk about real quickly is a molecule that's known as adenosine triphosphate, sometimes known as the molecule of ATP. This molecule, again, is still built from nucleotides, so you'll notice the sugar. And if you notice, it has an OSE ending, so that's the sugar. This right here is the base. And then notice over here, we've got 3 phosphate groups. And so this molecule right here, adenosine triphosphate, what it ends up doing is that it will go ahead here and it will donate these phosphate groups. And what that ends up doing is that this will go to some sort of cell, and that's going to go ahead and it's going to energize that cell. And so ATP is really, it's going to be energy at the smallest level for a cell. And this again happens to be the other form of nucleic acid. Okay? So anyways, like I said, what I'm now going to go ahead and do is I'm going to stop the PowerPoint here, and I'm going to continue with talking about some of the energy properties and then go through some enzymes and talk about the pH scale. Okay, so feel free here again to take a look at the previous video and just kind of go through that organic chemistry there. And let me know again if you happen to have any questions. Okay? Talk to you later. Bye-bye.